Prokaryotic gene regulation is much simpler. Remember that prokaryotes are bacteria. They're single-celled organisms. Why is gene regulation in eukaryotes more complex than in prokaryotes? This is because of the way that genes are expressed in a multicellular organism. Complex gene regulation in eukaryotes is what makes differentiation and specialization possible. In other words, the same DNA can code for different expression in different types of cells. Consider the following. Genes that code for liver enzymes are not expressed in nerve cells. Keratin, an important protein in skin cells, is not produced in blood cells. Cell differentiation requires genetic specialization, yet most of the cells in a multicellular organism carry the same DNA in their nucleus. What controls the development of cells and tissues in multicellular organisms? As an embryo develops, different sets of genes are regulated by transcription factors and repressors that bind to DNA. These, in turn, regulate transcription and the production of RNA molecules. Regulation of both DNA and RNA in this way helps cells undergo differentiation, becoming specialized in structure and function. The work of American biologist Edward B. Lewis made it clear that a set of master control genes known as homeotic genes regulate body parts that develop in specific parts of the body. The molecular studies of homeotic genes show that they all share a very similar 180 base DNA sequence, which was given the name homeobox. Homeobox genes code for transcription factors that activate other genes that are important in cell development and differentiation. In flies, a group of homeobox genes known as Hox genes are located side by side in a single cluster. Hox genes determine the identities of each segment of a fly's body. What section of the bodies of flies and mice is coded by the genes shown in blue? The back of the body, posterior portion of the abdomen of the fruit fly, and rump of the mouse.